This is lecture 22 on urinary system, part 2. If we look back to lecture 21 in urinary system, part 1, we covered the overall functions of the nephron in that it performed filtration, reabsorption, and secretion. We started with filtration and we'll continue with that here. We talked about the physical barriers affecting filtration, namely the fenestrated capillary epithelium, the basal lamina, and the Bowman's capsule epithelium or podocytes that affected the selectivity portion of filtration. But remember that for filtration to occur, we need to have two things. We need to have selectivity and filtration pressure. When looking at the factors that control glomerular filtration, we're looking specifically at factors that affect filtration pressure. Now, these factors, there are 4, are all going to be different forms of pressure in different directions. The first, and the one that contributes the most to this pressure, is going to be the glomerular hydrostatic pressure. If we remember, hydrostatic pressure is the pressure of a fluid pushing against the walls of its container. Here, this is looking at blood pressure in the glomerular capillaries that favors pushing water and solutes out of the plasma and into the filtrate. So when discussing this, I'm talking about moving from the capillaries and into the capsule. This averages about 50 millimeters of mercury a day. The next is capsular hydrostatic pressure. This is in direct opposition to the glomerular hydrostatic pressure. This is going to be the pressure of water pushing from the capsular space into the glomerular capillaries. Now, this is going to be far less than the glomerular hydrostatic pressure at only 15 millimeters of mercury. The other two pressures are going to be involved in osmotic pressure. We have blood colloid osmotic pressure, which is the pressure due to proteins in the solution. This draws water out of the filtrate and into the plasma, so it's opposing filtration. This is going to average about 25 millimeters of mercury. Then we have capsular colloid osmotic pressure. Remember, there's few, if any, plasma proteins that enter into the capsular space, so we have this at 0 millimeters of mercury. The only time this might actually change is if there's damage where excessive plasma proteins are going to enter into the capsular space, so the person would be sick or injured. This is a diagram illustrating the different overall pressures that we discussed. So take note of glomerular hydrostatic pressure being the largest at 50 millimeters of mercury. There's going to be a lot of plasma proteins in the actual blood, so this favors water movement from the capsule into the capillaries, and this would be blood colloid osmotic pressure, which opposes the hydrostatic pressure. Then we have capsular hydrostatic pressure. This is going to be the same concept as the glomerular hydrostatic pressure, but just in reverse. Remember that we have liquid in both containers that are pushing up against the space. Finally, we have capsular colloid osmotic pressure, and this is usually 0, so it's not really factored in unless the person is injured or sick. This means the total sum and net filtration pressure is going to be 10 millimeters of mercury, and this favors filtration from the actual capillaries and into the capsular space. Now, the careful regulation of the mean arterial pressure and associated cardiovascular mechanisms along with the renal mechanisms exists to ensure that this net filtration pressure is about 10 millimeters of mercury. Increased net filtration pressure could result in too much fluid leaving the circulatory system, and decreases in net filtration pressure result in poor removal of waste. So now we're going to look a little bit more at how the kidneys work with other systems of the body to ensure this filtration works as is intended. Now, as blood enters the afferent arteriole, about 1/5 of the plasma is going to enter into the tubular system of the nephron. This portion is called the filtration fraction. Now, the amount of fluid entering the nephron per unit is called the glomerular filtration rate, and that's what we're looking at here. The glomerular filtration rate averages about 125 milliliters per minute or 180 liters per day. Now that's a lot of filtrate, a lot of fluid. So why don't we urinate that much? Because about 99% of it is reabsorbed into the system. Now, This is influenced by two major factors: the filtration coefficient, which is controlled by the fenestrations and podocytes that we've already discussed, and the net filtration pressure, which is going to be due to blood pressure. Now, when looking at the regulation of the glomerular filtration rate, we have two interacting levels that help to stabilize it. One, which is autoregulatory at the local level, and a second, which is going to be a central regulation with regards to the central nervous system and the rest of the body. We'll start with the autoregulatory level. Autoregulation at the local level happens with two major mechanisms: the myogenic response and tubuloglomerular feedback. The myogenic response is going to be smooth muscle-based, whereas tubuloglomerular feedback uses two mechanisms within respect to the macula densa and the juxtaglomerular cells. We'll discuss those now. When looking at local regulation of the glomerular filtration rate, we're going to start with the myogenic response. The myogenic response is a lot simpler than the tubuloglomerular feedback. Here we can notice blood flow is flowing through the afferent arteriole. Now, the myogenic response, this response is an intrinsic ability of the vascular smooth muscle of the afferent arteriole to respond to a pressure change. So it's responding to stretch, much like blood vessels do throughout the body. Now, as blood pressure increases, the increased stretch of the walls of the afferent arteriole cause the smooth muscle to reflexively contract. So as it stretches, it will respond by contracting. This is going to cause vasoconstriction. The decreased vessel diameter will thereby increase resistance. Remember, as we increase resistance, this will slow down the flow rate, thus maintaining a consistent net filtration pressure. Now, if we flip it, if we decrease the blood pressure to where there's less stretch put on that afferent arteriole, we get a similar response, just opposite. So the decreased blood pressure is going to remove those contractions and cause the resulting diameter of the afferent arteriole to relax, thereby vasodilating and increasing flow rate. This increase in flow rate will thereby again stabilize the glomerular filtration rate based upon the overall blood pressure of the system. Now, we've covered myogenic response, which is a very kind of simple mechanism in that it's just about the stretch of the blood vessels and then the constriction of the blood vessels. Tubuloglomerular feedback is a little bit more complex and involves several different mechanisms. Now, this tubuloglomerular feedback is a local signaling mechanism in which fluid flow through the tubule feeds back to regulate the glomerular filtration rate in Bowman's capsule. So it's important to remember that we've demonstrated, or rather shown you, the nephron being kind of spread out. You have to remember that it's actually folded back on itself. And this gives rise to a structure called the macula densa. The macula densa are cells in the ascending limb of the loop of Henle and in the distal convoluted tubule. You have to remember this area is actually going to loop back around next to the renal corpuscle, as I'll show you in the next diagram. The macula densa is going to actually have cells. It's going to be a specialization of cells in the tube that are sensitive to sodium, to salt, sodium chloride. This activates the triggering of these macula densa cells are thereby going to activate the JG cells in the juxtaglomerular complex. The JG simply stands for juxtaglomerular. This will cause a decreased resistance to blood flow in the afferent arteriole by activating the mesangial cells. So the macula densa is going to be a sensory receptor cell in the ascending limb of the loop of Henle, or the distal convoluted tubule, sensitive to salt. So it responds to salt levels. Now, the juxtaglomerular complex is going to be a junction of the afferent arteriole and the distal convoluted tubule. So it's right where they meet. There's going to be a thickening area of cells that would eventually extend into the macula densa. These cells are actually going to secrete renin, which is a hormone that is involved in the overall regulation of glomerular filtration rate and blood pressure, etc. We'll get to that a little bit more later, but this is going to be released when glomerular blood pressure falls. The mesangial cells are going to be inside the glomerulus itself, and these are supporting cells. They lie between adjacent glomerular capillaries, and they help control capillary diameter and rate of blood flow. Now let's take a look at these three structures. Here we can see a diagram. Note in the top diagram, the distal convoluted tubule wraps up and around directly next to the renal corpuscle. It's going to meet where the afferent arteriole and the efferent arteriole are. So the cells inside the distal convoluted tubule are the macula densa. From there, we have where they meet that looks like a small triangle. We can see that is clearly labeled the juxtaglomerular complex. So the macula densa is actually going to sense these overall changes and transmit that information to the juxtaglomerular complex. The juxtaglomerular complex is going to initiate a lot of these responses. The mesangial cells, like I said before, are going to be adjacent between two capillaries in the glomerulus. Now, how do these mesangial cells work is if they relax, they actually make the blood vessels smaller. If they contract, they shrink down. So mesangial cells are going to be very vital in the actual regulation of this overall blood flow. Now we can actually put some of this to work with understanding the basis for autoregulation in the kidneys. So we'll start with homeostasis, where we have a normal glomerular filtration rate. From here, let's disturb the homeostasis and increase the glomerular filtration rate. If we increase it beyond that normal amount with the normal pressure of 10 millimeters of mercury, we're going to have an increased filtrate and increased urine production. So our body starts going to action. Autoregulation kicks in, and we have a constriction of the afferent arterioles via myogenic response, a relaxation of the mesangial cells, which is going to be done through tubuloglomerular feedback, and a relaxation of the efferent arteriole as well. From here, these three mechanisms are going to go forth and cause a decreased glomerular blood pressure. Now, if this was sufficient, then we can restore homeostasis and we're good to go. But in some cases, if that wasn't sufficient, we move on to the central nervous system and central regulatory mechanisms. Now, I've talked about and covered the autoregulatory levels, specifically dealing with the myogenic response, which deals with obviously smooth muscle constriction, tubuloglomerular feedback, which is going to use the macula densa, juxtaglomerular cells, and then the mesangial cells to reduce overall blood flow rate, but the tubuloglomerular feedback will feed into the next portion, which is going to be central regulation, if our autoregulatory mechanism is not enough to slow down glomerular filtration rate. So looking at the endocrine response, there are important hormonal pathways that influence the glomerular filtration rate. The most important of these is going to be the renin-angiotensin-aldosterone pathway. Now this is going to be initiated through tubuloglomerular feedback, but only if the glomerular filtration rate is going to be too high to where we can't regulate it with just our basic myogenic response. So it's going to be initiated through an autoregulatory mechanism and then feed into the larger endocrine system at central regulation. So they kind of work together. But we'll talk more about the renin-angiotensin-aldosterone pathway in subsequent lectures. The other response is a neural response. Glomerular filtration rate can be modified through actions of sympathetic neurons of the autonomic nervous system that innervate both afferent and efferent arterioles, causing vasoconstriction in a greater capacity. Now, if systemic blood pressure drops dramatically, say we're suffering from dehydration, we're bleeding out through a blood hemorrhage, etc., we have powerful vasoconstriction that will reduce the total renal blood flow. So reducing blood flow to the entire kidneys, thus decreasing glomerular filtration rate in an attempt to reduce fluid loss via the urine. Now that we finish with filtration, we get to talk about reabsorption and secretion. Now, reabsorption occurs throughout the actual tubule, but it primarily occurs in the proximal convoluted tubule. Reabsorption in the proximal convoluted tubule accounts for almost 99% reabsorption of glucose, amino acids, a bunch of other organic nutrients, ions like sodium, potassium, bicarbonates, magnesium, phosphates, sulfates, and of course, it's water. Water is going to be reabsorbed throughout, and there's a huge overall mechanism to involve water reabsorption into the actual capillaries, where water moves from the actual tubular fluid into the peritubular fluid. Here we can see a diagram of the proximal convoluted tubule looking at reabsorption specifically. Now, reabsorption requires the movement of materials, as seen here, where we see glucose and other organic molecules moving through, water, and then ions. It's going to move from the tubular lumen across the epithelium of the renal tubule and back into the blood. So it's going from the actual nephron tube into the blood vessels or capillaries. Now, this transport can occur via diffusion. It can occur via different channels that are open, or transport proteins. And this is all dependent upon what is being transported. Because some reabsorption relies on transport proteins, the process of reabsorption is subject to the same limitation as other transporters, to where it is saturatable. What this means is that there's a saturation limit or a transport maximum. Once we reach that limit, we can't move anymore. So we're capped off at how much we can actually move. Now, this is why uncontrolled diabetes mellitus results in glucose in the urine. And this is because the glucose transporters are saturated and we can't reabsorb any more back into the system because there's so much in the fluid. Secretion, remember, is the opposite of reabsorption. Now, secretion, as we've talked about, is the movement of water and solutes from the actual peritubular fluid and into the tubular fluid. So it's moving from the blood vessels and into the renal tubule. Now, this is the combination of reabsorption and secretion in the distal convoluted tubule alters the solute composition in the tubular fluid. It is important to understand that secretion primarily occurs in the distal convoluted tubule, but it does also occur throughout. Now, sodium ions are going to be reabsorbed in exchange for potassium ions. Hydrogen ions are secreted in exchange for sodium ions, and this is done to increase the pH of body fluids. And the carrier proteins also secrete toxins or drugs. Here we can see a diagram of the distal convoluted tubule and the exchange that actually happens. Remember that everything in this with respect to secretion is going to be about exchange. So we're trying to get rid of potassium ions in exchange for more sodium ions. We're getting rid of hydrogen ions in exchange for sodium ions, and we're using transport proteins to eliminate toxins and drugs. Remember, the transport proteins can be saturated, so there's a certain timeline where we actually can eliminate toxins and different drugs. So that's why the more you ingest of a toxin or drug, the longer it will take to eliminate from the system. Now, it is important to understand that secretion largely depends upon this membrane transporter system that generally requires active transport mechanisms. So it uses ATP to push this out of the system. In general, secretion, why it's important is to permit the enhanced excretion of a molecule. That's why secretion is so important in the body. So we've talked about filtration, we've talked about reabsorption, we've talked about secretion, and this is to eventually get to excretion. Excretion is where we actually finally leave the nephron, and what we have is filtered blood that's going to then be excreted. So this is going to eventually become urine or the renal excretion. Now, how we actually do this in the body, in general, the body has to take in the same amount of water that it gets rid of. So we ingest water via the digestive system, unlike other animals, but we also take a small amount through glucose metabolism, as we can see here. So this is simply looking at the fluid balance throughout the day. We get a certain amount from food, we get a certain amount from liquid, and then we get a certain amount as a metabolic waste product when we perform catabolism or any sort of aerobic respiration. Now, through elimination, we eliminate most of it through urination. Next is just through evaporation at the skin. Remember, the skin is a semi-permeable membrane. It's pretty water resistant, but it still does have some water loss. We have some evaporation at the lungs that occurs just from breathing, and we have a minor amount that is lost in the feces. This is showing you an overall diagram kind of flow chart of how we would balance this out throughout our digestive tract. So you can see dietary input includes the food and drink. We have different digestive secretions that actually use some water or put some water back into the system. Things like saliva, our gastric secretions from our stomach, liver, bile, pancreatic juices, and then intestinal secretions. Then finally, we have colonic mucus secretions. Now, most of this actually gets reabsorbed back into the system, primarily via the small intestine, but also through the colon as well. So this is going to end up balancing out to where we lose a very minimal amount of water via the feces. When looking at the cellular level, it's important to go back to our basics and understand that we have our cell, which is a membrane that has fluid inside of it, and this is the intracellular fluid. And then we have the fluid that is surrounding the cells, and this is the extracellular fluid. Now, most of our liquid is actually going to be in the extracellular fluid, but we have an overall balance of water throughout the system to make sure that we're not overloading the cells to where we remain, again, isotonic. Now, we have a certain amount of water that is absorbed across the digestive epithelium and a certain amount of water that is lost in the urine. As long as we're equally exchanging water, we don't really lose too much size of the cell. However, in some cases, we're not replacing water if we actually have dehydration. So we start with a nice balanced scenario, as we can see up top, where intracellular fluid and extracellular fluid are balanced, and the two solutions are isotonic. Now, let's say we work out, we sweat, but we're not drinking any water, we're not drinking anything else, we're not taking anything in, and we stay like this. The water loss from the extracellular fluid will decrease its volume. This makes the solution hypertonic, so it's more concentrated. We lose some salt, but we're losing more water than anything. So because of this, understand that the solution balance between extracellular and intracellular fluid are now out of balance. So an osmotic water shift from the intracellular fluid moves to the extracellular fluid, thereby restoring an osmotic equilibrium. However, we have now decreased the overall volume of intracellular fluid, and we're making our cells more concentrated. Now, there is a problem with this over time. Why is the regulation of this so important? Due to 4 factors. First, sodium is critical for many transporters and is a key determinant of body fluid osmolarity. If this balance ever gets shifted, we could have an overall error in function. Number 2, potassium and calcium have critical roles in muscle contraction, including the heart and other processes. We have hydrogen ions as well as bicarbonates that are going to actually be very important determinants of body fluid pH. And finally, if the extracellular osmolarity changes too much in any of these areas, cells may be damaged. Remember that increased osmolarity causes cells to shrink. Low osmolarity can cause them to swell. We remember this back from our concepts on diffusion and osmosis. So keeping an isotonic solution is important, but we also need to have a certain balance in that solution itself to make sure that it's not too concentrated in damaging some of the internal structures inside of the cell. Now, how does the body accomplish a lot of this regulation? It's done through a number of systems that are integrated together. Remember, while we're breaking down the body into its constituent parts, the body never works independently. It's always multiple systems working together to maintain everything in the body. In this case, body fluid and electrolyte homeostasis. These systems include the renal system, respiratory system, digestive system, cardiovascular system, and the endocrine system, where we can have ion absorption across the epithelial lining of the small intestine and colon. We have ion reserves in the skeletal system, and we have ion excretion through sweat glands and kidneys in order to balance the system out. And we can control and regulate all of those areas to maintain a balance between our intracellular fluid and our extracellular fluid.